peas, mayonnaise, and pizza. These are three things that should not be at the same place at the same time, unless you're this one guy on Twitter. How is it possible that individuals can have such different responses to the same piece of food? Your taste in food has many influences: who you grew up with, where you've lived, and even your genetics. All these factors come together in one place, the only place where these preferences could live, your brain. It has been an open question exactly how these preferences are stored in the brain, and how the encoding of visual information varies between subjects more generally. I'm Leila McKinney, and this summer I worked with Dr. Leila Wehbe and Dr. Michael Tarr through the undergraduate program in neural computation. My project, Your Brain on Pineapple Pizza, introduces a framework to explore and interpret differences in brain encoding models trained on different subjects. To explain our methods, we first ought to discuss the project's background in both computer vision and neuroscience. Simply put, the problem of brain encoding asks us how the human visual system takes complex, real-world stimuli and reshapes them into flexible internal symbols. To understand and model this process, cognitive neuroscientists have used fMRI responses to build a map of general concepts in a brain. These include regions such as the fusiform face area, associated with face recognition, and the parahippocampal place area, associated with natural scenes. Recent work has expanded these techniques to predict the functional correlations between image features and neural activation for a subject with millimeter-level accuracy. This progress comes largely from neuroimaging datasets and machine learning models for computer vision. The natural scenes dataset, released in 2021, contains thousands of color images paired with high-resolution fMRI responses across 8 subjects. Released the same year was OpenAI's CLIP model. Trained on over 400 million image caption pairs, CLIP allows image data to be transformed to a much smaller space, organized by image semantics. In my project, I trained 8 brain encoding models, one for each subject. Each model predicts a neural response to arbitrary images. I used CLIP to predict image features, then a linear probe to map these features to neural responses. Shown on screen are the dimensionality-reduced features of weights for the different voxels of the food region in all subjects. Similar colors indicate voxels that are selective of similar image features. As you can see, the functional organization of the different brains appears subtly different. We then employ these encoders to discover images that are characteristic of each subject. That is, images that are predicted to have high mean activation for one subject, but lower activation for others. We do this in 2 ways. First, we sort a large dataset of food images according to this heuristic. Second, we use this as an objective function for brain-guided image synthesis. Together, these techniques enable us to identify clear food palettes for subjects that we missed had we optimized according to the response of only the individual or population. Brains are far more alike than they are different. However, it is in these differences that lies selfhood. This work explores subject-wise differences in the brain encoding of visual semantics of food images. Applications include improving our understanding of neurodivergence, as well as the synthesis of calibration images to fine-tune brain encoding models to novel subjects. Thank you.